welcome to this video. So in this video we are going to talk about the cell and the organelles that are found in this cell, and also we are going to touch briefly on what is membrane transport. Okay, so for viewers on YouTube, make sure you like and subscribe, and let's get right into it. Okay, so this is a brief content overview of what we are going to cover. We're going to talk about the cell structure, the structure of the plasma membrane, the cell organelles, the inclusions that are found in these cells as well, and also we're going to touch on the cytoskeleton of this particular cell. All right, so cells, we by now would have learned a lot, like starting from grade 8, for those in the old curriculum. We've been talking about cells being the structural and functional unit, the most basic structural and functional unit of a living organism. What that statement simply means is that at the where life begins, okay, where you can start counting life from is basically that single cell. Okay, that's where all the processes necessary for life, that's where they occur in a cell. We actually have organisms that are unicellular. This means that it's a single cell, but it's an organism on its own. Okay, so complex organisms are the ones that are multicellular. They're going to contain a lot of cells in them. That's where the word multi is coming from. While simple organisms are going to be unicellular. That means you might find an organism, like I said, that only has one single cell. Okay, so there are two classes of cells that are of particular interest to people who study these cells. There are cells that are known as eukaryotic and prokaryotic cells. Okay, so eukaryotic cells, these are cells that you find uh, mostly in the complex organisms that are multicellular. For example, in plants, in animals, you're going to find what are known as eukaryotic cells. But prokaryotic cells, on the other hand, these are cells that are usually found in simple organisms and are usually unicellular. For example, bacteria and viruses, all of those are prokaryotic cells. All right, so the different cells in the human body that you're going to find are differentiated based on their functions. Okay, so although they originate from a single point of origin, you're going to find that they end up having different specializations, specialized for a particular function. Okay, so the process of adopting a specialized structure for a particular function is what we're calling differentiation. Okay, for example, the development of a zygote into a full human, you're going to have cells there differentiating into, let's say, for example, epithelial cells that are going to be lining the skin. You're going to find liver cells. Although you are going to see that a zygote is a unicellular structure. That means that's one cell, and then you're going to have many cells arising from this zygote. So there's going to be cell division. You're going to have two cells, 4 cells, 8 cells, 16, and then at the end of the day, you have a full human being. So, how do these other cells come about that have different functions, like epithelial cells, like hair cells? You have cells of the eye. What is that process called by which you're having those different cells from just one cell, this zygote? That's what we're calling differentiation. All right. So, uh, we can move on. So cellular structure, although cells might be different, like in the human body, for example, there are cells that are found in the arm, in the skin, cells that are found in the nails, cells that are found in the liver, like I said. They have different structures and different specializations, like I said, but they have a similar basic structure, and uh, that's what we are going to offer. Alright, that's what we're going to explain in this particular tutorial. So the basic structure that is similar to the structure that is similar in all of the cells that you're going to see is what we're going to explore here. All right, so there are two basic parts of a cell, one being the cytoplasm and two being the plasma membrane. So the basic parts, cytoplasm and the plasma membrane. So in the cytoplasm, it surrounds the nucleus and all other organelles and is the site for many enzymatic reactions of the cell. Okay, so in the cytoplasm is where you find everything else in the cell suspended, and then all of that is held together, or rather, all of that is kept from the outside environment, is barriered from the outside environment by the plasma membrane. So the plasma membrane separates the cytoplasm from its extracellular environment. As you can see, the plasma membrane here is separating the cell from the outside environment, and the cytoplasm on this other hand is what we are seeing, that we have many organelles uh, being found in the cytoplasm there. The nucleus, as being an organelle as well, is being found in the cytoplasm, or the cell membrane is separating the inside and the outside of the cell. All right, so we are going to explore a little bit more of this plasma membrane, or the cell membrane. Okay, so this is basically a physical barrier from separating the cell from its outside environment and its inside environment, because inside the cell you're going to have different conditions that are required for different subcellular processes. So that is going to make sure. Okay, so I was saying it forms a physical barrier between the internal and external environment of the cell, because there are different processes that are happening between these two sides. The internal environment and the external environment have different types of processes in them. So we need this inside of the cell to be barriered from the outside, just so that those processes can occur in a well buffered environment. For example, you need the pH of the inside of the cell to be maintained at a certain level. You need the concentration of different ions and other substances to be maintained at a particular level, so that the cellular processes of this particular cell are maintained properly. Okay, so it is composed of phospholipids, proteins, cholesterol, and oligosaccharides. Okay, so uh, the phospholipids make up what is known as a phospholipid bilayer, stuff that was explored extensively in first year, which we are not going to really take much time on. But the proteins that we are of particular interest here are the what are known as integral proteins and structural proteins. So structural proteins help in maintaining the structural integrity of the cell. Okay, so they help maintain basically the shape of the cell, and they also function as receptors, for example. And they are also what are known as integral proteins. Integral proteins, these ones are going to serve mainly as ion channels for membrane transport. Okay, so membrane transport, because this particular bilayer is made of lipids, so you're going to find that molecules that are charged, for example, cannot easily pass through, cannot easily diffuse through, or molecules that are polar cannot easily diffuse through. So you're going to need some sort of help. Okay, they're going to need some sort of help to cross this membrane. So how does that occur? Through ion channels that are part of integral proteins, that are made up of these integral proteins. Okay, so let us look at how these occur. So transport across the plasma membrane depends on the molecule type. Like I said, fat soluble molecules will pass through the lipid bilayer through simple diffusion. And this is going to be easily done. Simple diffusion, as you can see here, the molecules that are fat soluble are going to be diffusing through the membrane just like that, as easy as that. But those that are fat insoluble, like polar molecules, for example, and molecules that are charged, these are uh, molecules that are not able to pass through easily. So they're going to utilize a channel, okay, and a carrier or a transporter. So the difference between a channel and a carrier or a transporter is that a channel is simply allows the movement, the passive movement, the passive movement of a particular molecule from the inside to the outside or from the outside to the inside of the cell, just acting as a passageway. Okay, while a carrier and or a transporter utilizes energy. Okay, so this is used mostly in active transport. As we are exploring the types of transport, we'll see exactly what that is about. So this is how membrane transport occurs in a nutshell. So to get a little bit deeper into that, we're going to talk about the types of membrane transport that we have. So firstly, we have passive transport and active transport. The difference between these two is simply the use of energy in active transport, while in passive transport, no energy is used. And the examples that fall under passive transport are diffusion, facilitated diffusion, and bulk flow. So we're going to talk about these in a little bit of detail. So diffusion, the same basic definition that we've always had: movement of molecules from a place of their a high concentration to a place of lower concentration. That's what we call diffusion. So the membrane uh, allows passive diffusion of molecules. There's also what is known as facilitated diffusion. So before we go to facilitated diffusion, the molecules that are able to passively diffuse through this membrane are those that are uh, those that are fat soluble or lipid soluble, because the membrane is a lipid layer, like we said, it's made out of lipids. So those that are able to diffuse freely through passive diffusion are those that are lipid or fat soluble. While in facilitated diffusion, we're talking about those molecules that need a channel. Those ones, although they are moving from an area of high concentration to an area of low concentration, they still need a channel because they might be either charged or they might be water soluble. Okay, so they might be polar, charged, or water soluble. For example, ions, you might find that they need this form of membrane transport known as facilitated diffusion. And then there is also what is known as bulk flow. So this is simply the movement of a lot of substances in or out of the cell. For an example of bulk flow here, you might have filtration, where you are forcing a lot of water molecules to leave the cell at a particular time. For example, the processes that happen in the kidneys during filtration rather. Okay, so that will be explored extensively in physiology. All right, and then moving on to active transport. Active transport is segmented into two. There is primary active transport and secondary active transport. Okay, and another type of active transport is vesicular transport, which is sometimes referred to as a special type of active transport, but we can consider it as a third type. It doesn't really matter. All you need to know is what is happening in this particular type of transport. So, for primary active transport, you're talking about a type of active transport which utilizes ATP or energy directly. The form of energy currency in the cell is ATP. So, in primary active transport, you're using that energy directly. And by direct, we mean this ion channel that is here utilizes an ATP molecule to move these molecules in and out of the cell. But in secondary active transport, you're using the energy indirectly. So, what is happening basically is if you are moving a molecule from the outside to the inside of the cell, for example, using ATP, another molecule can use that molecule as the energy or the gradient that is created by moving that first molecule from the inside to the outside or from the outside of the inside to the cell. You can utilize that gradient created to move another molecule. So it's more like you are riding on the energy that is already there. So it's like you hitch a ride, like let's say someone says, I'm going to town and they have a car. So you say, okay, since I'm also going to town, I can just hop on their car and go with them. Okay, that is basically what is happening in secondary active transport. You're using energy that was used by another molecule that is in its primary or its primary transport rather. Okay, so that's what secondary active transport is, utilizing energy indirectly. And thirdly, we have vesicular transport, which is also divided into two segments. We have endocytosis and exocytosis. So in vesicular transport, you have formation of what are known as vesicles. Okay, cells are able to release or uptake materials in the form of vesicles. For example, vesicles that are released at the neuromuscular junction in the form of SCH vesicles. Those are what we are calling as vesicular transport, and the cell can either take in these vesicles or uh, uh, export material through these vesicles. So if it takes them in, you're calling it endocytosis. And then if it's taking them out, it's called exocytosis. And then under endocytosis, there are also subsegments of endocytosis, examples of endocytosis, which are including phagocytosis, which is cell eating. We usually talk about this when you're dealing with immune cells. Pinocytosis is also called cell drinking because it is uptake of vesicles that contain extracellular fluid, basically. But phagocytosis, that extracellular fluid or that material that is being taken in by the cell contains uh, contains substances. For example, it might have a host cell, which might be a bacteria, a pathogen, or anything else. But in pinocytosis, it's only the extracellular fluid itself. So this one will be called cell eating, can also be referred to as cell eating, and this one can also be referred to as cell drinking. Okay, and then we have receptor mediated endocytosis. Receptor mediated endocytosis is where uh, we have use of certain signals. Okay, so a certain chemical is going to bind to a receptor on the cell membrane that is going to cause that part to form a vesicle that now curves in, and then it will be taken in by the cell. For example, a a hormone. A hormone is one of those ligands that might have to bind to the certain receptor on the cell membrane, and then when it binds to that receptor, it's going to cause the process of endocytosis to be started for that particular segment of the cell membrane. All right, so that is that on membrane transport. So moving on, we are going to talk about the cytoplasm and the organelles that are found in this cytoplasm, and then we'll call it a day from there. We're going to try to be as quick as possible because most of this stuff is stuff that you have already done since grade 8, basically. So it's the same thing. So the cytoplasm consists of a fluid component called the cytosol, which is made mostly of water and dissolved elements. These dissolved elements might include oxygen, carbon dioxide, electrolytes, metabolites. Waste products are also included there, and the enzymes are also present in this cytosol. Okay, so the cytosol also includes metabolically active structures which are known as organelles. So this is the definition of an organelle: metabolically active structures that are found in the cell. And then there are also protein components, which are cytoskeleton, which we'll explore a little bit further. And there are also what are known as inclusions. Inclusions are glycogen granules, lipids, or pigments. Okay, so these are okay, and those are the inclusions. So uh, the organelles in the cell that we're going to talk about, like I said already, these are the metabolically active structures that may be membranous or non-membranous. That means we might have membranes separating them uh, from the cytosol environment. So there are structures that also need a microenvironment around them that is supposed to be specific depending on the function of that particular uh, particular organelle. For example, what is happening in the nucleus shouldn't happen elsewhere, so you're going to make it membrane bound. That's what we mean. Okay, but some of them are non-membranous, and we're going to see which ones are which. So for membrane-bound structures, organelles, we have the rough and smooth endoplasmic reticulum, the Golgi apparatus, the lysosomes, the peroxisomes, and mitochondria, including vesicles for transport, secretion, and storage of cellular components. Those are structures that are membrane bound. But for the non-membranous organelles, we have ribosomes and the cytoskeleton. These ones are non-membrane bound. So please make sure that you have these at your fingertips. Okay, so we're going to start looking at these organelles one by one, and we just cruise through them because most of them are stuff that we already know, and their functions are basically stuff that is well known. So the nucleus, we know that it controls all the cellular activities, and it does so through the processes of uh, gene expression. You have DNA being involved there. So we know that for most cellular functions, a certain protein has to be involved. Where is this protein coming from? It is coming from a specific sequence of amino acid that is encoded on the DNA that is in the nucleus. So that process starting from DNA, RNA, and finally proteins, and these proteins are going to be controlling various processes that are going to be occurring in the cell. That's how the nucleus controls uh, different activities in the cell. So uh, that is one of the most important functions of the nucleus, while the other is production of ribosomal subunits. So the ribosomal subunits are made in the nucleolus, and they are exported to the cytosol, and where they're going to be they're going to be arranged into the full ribosome that can now undertake the process of uh, final protein assembly. Okay, so that is basically what's happening there. So the components of the nucleus has a nuclear membrane, okay, which is a membrane, like we said, we classified it under membranous membrane bound structures. So that means it has a membrane around it, and it's called a nuclear membrane. And it's a double membrane with perinuclear space that allows with selectivity exchange of material in both directions. So what we are saying is that it has these pores, which are known as nuclear pores, and it has what is known as the perinuclear space, which is the space that you're seeing right there. And then inside there you have the nucleolus. Okay, and then other stuff that you find in this nucleus are what are known as nucleoproteins. Nucleoproteins, these are proteins that are found in the nucleus basically. So nucleoproteins, proteins that are found in the nucleus. And an example of these are histones, and some of them are non-histones. So histones form a protein core around which the DNA is called to form nucleosomes. We'll explore this in a bit. While those that are non-histone proteins, they include all the enzymes for the synthesis of the DNA and RNA and other regulatory proteins. So we know that during the process of DNA replication and RNA formation and the like, we have those enzymes, helicase, DNA polymerase, what, all of those are non-histone proteins that are found in the nucleus. And we know that enzymes are proteins, so that's what exactly we are talking about here. And other regulatory proteins are also found like that. So this is an example of a nucleosome. A nucleosome is histone proteins, and the DNA is just wrapped around it like that. So we have H1 histone here and H2A, H2B, different type of histones there. Nothing much to worry about. All you have to know is that uh, when you have DNA wrapping around a histone protein like that there, you're going to have what is known as a nucleosome. Okay, so the outer membrane of this nucleus, the new membrane that we talked about here, is continuous with the rough endoplasmic reticulum. This is because as you have gotten your mRNA here, okay, transcription, this process of transcription, translation has to occur outside the cell. So the mRNA has to pass outside here, and then here where you have the ribosomes that attach to the endoplasmic reticulum, calling it the rough endoplasmic reticulum in this case. They're going to work on that information in the mRNA and form a protein that is going to be folded here, and it's going to be uh, processed later in the Golgi apparatus. That's basically what's happening. So the outer membrane is continuous with the endoplasmic reticulum. That's what we are trying to show here. Okay, so in the DNA, we also have what is known as chromatin. This will be very important as you are starting cell division and going forward. So chromatin is basically a structural form or structural arrangement of DNA. In non-dividing cells, DNA exists as fine strands called chromatin. So it's going to be one strand that is going to be very fine, uh, like this. So that's like a strand of DNA that is very fine and is not coiled at all. That's what we mean by fine. By fine, we mean it's not coiled. It's called chromatin. So that DNA as it exists in fine strands is called chromatin, and it consists of the DNA and its associated proteins known as histones, the ones that we are from talking about. So the DNA is of two types. The chromatin is of two types. It has heterochromatin and euchromatin. Okay, so before we get to this, let's just explain a bit on how we have DNA moving to chromatin. How does DNA form chromatin? So DNA, we know that it's a double-stranded helix, a double helix that we usually talk about. DNA double helix. So that double helix is going to wrap, that strand of double helix DNA is going to wrap itself around histone proteins, and those are going to form nucleosomes. Then these are going to further clamp together. These are going to further clamp together, and they're going to form a fine strand of chromatin that we are seeing here. This fine strand of chromatin can either exist as heterochromatin, which is tightly packed, or euchromatin that is still in fine strands. Okay, and then when you pack this further, you're going to get what is known as a chromosome. Okay, chromosome, that's an entire chromosome that is duplicated with a centromere there, that we're going to explore further when we're talking about cell division. Okay, so this is where now the euchromatin and the heterochromatin comes in that I just explained. If it is supercoiled and electron dense, it's going to be called heterochromatin. While if it's euchromatin, it's electron lucent. That means there are not much electrons, and it's going to be in fine strands. And this one usually exists when the cell is in active uh, protein synthesis or protein translation. All right, so the nucleolus, like we said, the nucleolus is the main site for RNA synthesis and ribosomal assembly. Transfer RNA is also processed in the nucleolus. That's basic. I think we already talked about this. All right, so the ribosomes themselves, these are small electron dense particles, also electron dense, that are composed of four different segments of ribosomal RNA. So these are assembled in the nucleolus, and then their main function is basically to produce proteins. They read the information on the mRNA and assemble. Uh, they're going to assemble the monomers that are required for forming uh, proteins, which are the amino acids. Okay, that's basically what they do, and then uh, they have a small subunit and a large subunit, which will be further explored when you're doing biochemistry, protein translation, and everything. Okay, and then the endoplasmic reticulum, of course, these ones are involved in protein synthesis and uh, uh, lipid synthesis. So the protein synthesis is mainly the function of rough endoplasmic reticulum, while the lipid synthesis, the steroid hormone synthesis, and detoxification are the functions of the smooth endoplasmic reticulum. And the difference between the two is simply that the rough endoplasmic reticulum has ribosomes attached to its surface. These ribosomes are the ones that are doing protein synthesis. That's why the function uh, there correlates as well. All right, so moving on to the Golgi apparatus or the Golgi complex. These ones are a series of parallel stacks of flattened circules and associated vesicles. The main function of these Golgi apparatus is that they perform what is known as post-translational processing of proteins. That means packaging proteins for where they're going to be going, and packaging them based on their function, whether they're going to be remaining in the cell or they're going to be exported out of the cell, all of that. So these Golgi apparatus has what is known as a cis and a trans phase. A cis phase is where it faces the rough endoplasmic reticulum, where it receives the proteins from. It's called the cis phase, while the trans phase is where uh, it's facing the membrane of the cell, where it's going to be exporting from. So cis phase, cis side or cis Golgi is where it's receiving proteins from the rough endoplasmic reticulum. They can ask you an MCQ here that the cis Golgi always faces the rough endoplasmic reticulum, or essentially you need to know the orientation, cis and trans Golgi. Very important. Okay, and we also have mitochondria. Mitochondria are known as the powerhouse of the cell. We have had this definition since whenever. So we're going to make sense of this definition in biochemistry when we talk about how ATP is produced. So yes, it is the powerhouse because that's where ATP, the energy currency of the cell, is produced in this mitochondria. So this process is going to be explored further in biochemistry. And finally, we have, oh, not finally, but we have inclusions. Inclusions are non-metabolically active substances in the cell. And then these common inclusions include glycogen, lipid droplets, pigments, crystals. These are not stuff that is involved in doing any, physical process, or rather any metabolic process in the cell. They are just there, maybe being stored or as uh, products of some particular process. The examples are glycogen, for example. We have much of it in the liver cells, the muscle cells, the lipid droplets that are stored in the fat cells. We have pigments that might be found especially in the spaces of the liver cells as well. Uh, we have crystals. These ones might also be found in uh, in the kidney, for example. Yes, so that is the inclusions. In the cytoskeleton, on the other hand, which is the last cellular structure we're going to talk about, is a three-dimensional meshwork of protein filaments that are responsible for the maintenance of cellular morphology. So they help keep the shape of the cell and also its structural integrity. They provide mechanical strength, and they play a role in locomotion. Okay, and another important feature of the cytoskeleton is that it's important in forming mitotic spindles, which are important in the topic that is coming, cell division. Okay, so it has three major components, the cytoskeleton. So it's basically more like a skeleton for the cell. So like we say, these structures that you're seeing, mechanical support, keeping the shape, locomotion. You find that these are basically what almost the human skeleton does as well. So you can consider it as that for the cell particular. We have three major components of the cytoskeleton. We have thin filaments, intermediate filaments, and microtubules. So for microtubules, these are just tubular hollow structures that organize in a large arrays to form axonemes. So an axoneme is this particular uh, feature that we are seeing here, and you have tubular or hollow structures that are arranged in different orientation, like what we are seeing here, that makes up this particular axoneme. And then they are going to form different structures that are responsible uh, for forming other cellular extensions. Let's call them that for now, that are responsible for other cellular functions, like for example cilia and flagella. So for example, the cells that have cilia, at their core, they're going to have these structures that are made up of microtubules. Okay, and the proteins that make up microtubules are alpha and beta tubulin. So they originate at the centrosome, the microtubule at organizing center. That's basically what microtubules are. So the main takeaway here uh, from microtubules is that they form the core of structures like cilia and flagella. You're going to find the cells that we're going to be talking about in the subsequent lectures will have some of these specializations, and just know that at their core they have these microtubules. Okay, that's the main takeaway message there. For microfilaments, they're made up of actin filaments or thin filaments. These ones are mostly important in cellular movement. Okay, like for example, in the muscles, they're the ones that are responsible for sliding with thick filaments, and they produce muscle contraction. And also cellular motility as well. Like I said, movement of the cell is also a function of these microfilaments, and they also form a band just below the cellular membrane that forms a more like a supporting structure to help keep the shape of that particular cell. So, and I take home here microfilaments, very made up of actin filaments, and they are important for structural support and locomotion, basically. Then intermediate filaments, on the other hand, these are more stable than microtubules. They're more like in the middle of microtubules and microtubules and microfilaments. So they're more like in the middle, and they're made up of different protein subunits, and these ones are more stable. Okay, so they're more stable because they're made up of different protein subunits, and examples include keratin, neurofilaments, laminins, vermentin. These are you're going to be finding as we move in anatomy. You're going to be finding these. So for now, just take them as examples of intermediate filaments. That brings us to the end of this particular tutorial on the cell organelles and membrane transport.